Acid Journal for this fantastic opportunity to discuss with Dr. Arutha Kulasinghe. So we will have a chat, informal chat for the next couple of maybe 30, 45 minutes. And we will be happy to hear from you about your research and where you come from and where you're going with it. Again, the recent achievement that you got, the grant that we will talk and explain more about. All right, so why won't we go to the beginning, right? Give a bit of a background of how we all got here. Can you kind of give us and walk us through, give us a bit of a background of what has been your research journey to date? We'll be happy to hear about it.
Yeah, no, absolutely. Thank you for the invitation to talk about our research and where we've got to. It's it's great to be here, and yeah, looking forward to this discussion. So we got into this space a couple of years ago, very serendipitously. I did my PhD in sort of circulating tumor cells, trying to characterize them in the liquid biopsy space, which you are very much a thought leader in, and so we were very involved in that liquid biopsy space and the evolution of that. But sort of a piece that kept coming to me was in that you still need to characterize the tumor and the liquid biopsy at the same time. So in my postdoc, what I did was I sort of went quite deep into characterizing the tumor microenvironment, and some of the tools that were available at the time, very early on, sort of 2017, 18, were very, multiplex IHC, a couple colors. But we thought that the data we were getting from it was useful because for the first time we could start to, on the same tissue section, see multiple different markers, multiple different biomarkers. And so even though it was low plex at the time, it sort of planted the seed that, being able to characterize the tumor microenvironment in its totality is really important, and potentially important for immunotherapies, where we know that these patients tend to do well, a subset of patients tend to do well on immunotherapies, and trying to identify those patients, whether it's skin cancer, head and neck cancer, lung cancer, triple negative breast, IO therapies are, they're beneficial in a number of tumor types that lead to long-term outcomes and durable benefit, but identifying those patients is key. It's an area of strong interest in the liquid biopsy space, but also in the tumor microenvironment space. So, and then at the time, we started getting into a few of these newer technologies that were evolving. And so we were able to do, 30, 40, 50 markers at the same time on the same tissue at the protein level. But then the RNA came online. So that was super interesting. So we could look at serial sections of the tumor microenvironment from patient specimens, either surgical sections or biopsy sections. These are archival pathology specimens. We can go back in time, and we could characterize we could access the tissue block from a pathology lab, section it, and run the RNA and protein. And what we discovered was that using well-defined cohorts of patients where you have responders and non-responders to a certain IO therapy, we could ask the question, what in the tumor microenvironment, whether it was stroma or tumor compartments, either at the RNA level or the protein level associated with response to therapy. Very basic questions, but it was a lens of new discovery on the tissue, which we found was really informative. So that's where our labs now focus on in the last couple years, really doing a deep dive of the tumor microenvironment, both at the whole transcriptome level, but also at the sort of high plex protein level. So looking at over 100 different proteins with single cell resolution. And it's fascinating, like looking at these tissues where you have single cell readout for over 100 different proteins. You can start to see the cellular architecture. You can start to see, not just T cell infiltration, but you can start to see areas of resistance just based on the immune landscape, but then you can get deeper into that by looking at the metabolic data. So you're sort of layering these omics data sets together. So for us, it's a new lens on known biology, right? But we're trying to take sort of deconstruct this multi-omics cloud from patient samples. Yep.
Right. That's fascinating. So I'm part of the era that when we started, it was a bunch of epithelial cells a cell line that is sometimes 50 years old that you put on plastic dish and you try to knock down a gene or overexpress some protein and then try to deduce or get insightful information about what's going on inside the tissue. And people did get to many discoveries and insights throughout these decades, but it seems like there's a leap here getting to the fierce, complexity of the cancer tissue. So when was it, the point in time that you were like, okay, this is not enough. We need to get deeper to get those answers. So it's something that it's gradual, but it's also something that you need to decide with yourself, right?
Mm-hmm. Yeah, no, great question. So for us, obviously, a while ago, bulk was the approach, right? You mash a tumor up, you profile it for all the proteins, RNA, and you get a readout. It's an average of everything, right? And then single cell came out in 2011, and single cell is pretty much you're ordering the cells, or you're dissociating the tumor, and you can order the cells. You get a t-SNE plot, you can see all the cell types, but you sort of don't know where it's coming from, right? So for us, it was the aha moment when you could map that t-SNE back to the actual tissue, right? So you can understand where the cell type is in the position in which it's located in the tumor or the microenvironment. So we're not losing that, we're not losing any dimensionality there. So for us, we took that leap of faith, but we took it in that knowing that getting the tissue contexture is important. And we understand that this is a sort of long-term process, and understanding how to map these proteins and RNA in space is a process, and it's still early days, but we've sort of taken that leap of faith, knowing that we're inferring that anyway from single cell data, right? So I think and we can start to, we're not running hundreds of samples at the moment because the costs are prohibitive. But from the tissue microarray studies where we can put 90 cores on a TMA from 90 patients, those sorts of studies, and then we do whole sections from certain patients, we can see that getting that information directly off the tissue makes sense, because that's the context in which it's found, right? We're not inferring anything there. But yeah, I like to say we grew up in an era of spatial biology, and I'm probably lucky to grow up in that, just like you might have grown up in a previous sort of maybe single cell or bulk era. But it's sort of the new tool. It's a new lens, right, that we can look at tissue, and I think even looking at normal tissue is fascinating, because you can start to understand how tissue ages and things like that, right? Not just in the cancer context, but and
maybe also other pathologies. I mean, the normal, but also other pathologies of different tissues.
100%. I mean, we've looked at the lungs of COVID-19 patients using this approach, the hearts, the brain. So it's a methodology that it's tissue profiling, right? So you can apply it to any sort of methodology where you have tissue. Right.
That's really spot on, because I mean, we started cancer researchers, but those technologies could really get expanded to any type of tissue profiling. But let's really maybe focus about the recent grant or the award that you received. So you proposed to do something, and I, went through previous publications. So you're not specifically a skin cancer biologist, because you have a publication related to GBM and head and neck cancer. So what would you say drew you through the skin cancer field? Where do you think you can contribute? What did you propose, and what do you feel that we can gain from what you offered to do?
No, great question, and it's sort of it's where we've headed. So we initially, my area of research is head and neck cancer, lung cancers. That's what we really focus on. But the challenge with lung cancer is that when you get a biopsy sample, it's pretty much a fine needle aspirate, tiny samples, right? Head and neck, you can get more tonsillar, you can get a larger bit of architecture, but it's still invasive. So the reasoning to go into skin cancer has been that immunotherapies are effective in multiple tumors, right? Melanoma, head and neck, skin, lung, triple negative breast, renal, right? All these tumor types where you have immune responsive phenotypes. So our question was, skin cancer is easily accessible. Therefore, why don't we look at patients that had a complete response to IO? 2 years out, they're still good, and then patients that had a progressive disease to IO, and then at those extremities, right? Not the partial response or the stable disease. I think at the extremities, the complete responders and the progressive disease, when we look at those phenotypes, and skin you can biopsy over the course of therapy, right? So it's an easy tissue type to work on. So our understanding was, let's look at these two sort of discrete cohorts, look at the skin tumor microenvironment, and hopefully we'll learn something that's valuable to skin, but also potentially applicable to head and neck and lung. So that's been our approach here. And then we're running a study at the moment. The study that got funded is to look at the skin tumor microenvironment in patients that progressed and didn't progress on immunotherapy, but we get a biopsy prospectively even at cure. So multiple biopsies over the course of therapy, over the course of IO, and even at cure, we get a biopsy sample and at progression. So then we can look at the sort of the temporospatial changes in the microenvironment over time, which you can't do for lung cancer. You can't re-biopsy,
Right.
and neck is challenging. So that's
much
been our
more
reasoning.
Approachable, right?
Exactly. So we understand that it is a different cancer type, but we think the lessons we can learn from skin cancer may be applicable to head, neck, and lung.
Right, and it's not like we don't need those spatial distribution in those kinds of. So the unmet need is still huge, also in melanoma and other types of skin cancer. But and maybe the fact that it's melanoma specifically is one of the most immunogenic, and tumor microenvironment has such a major role played in it, could help you make predictions and also related to therapy, maybe.
Yeah, exactly. I mean, some of the work we're doing when we start to look at, over 50 or 60 proteins, or even the transcriptomic data, is we can identify the cellular neighborhoods within the tissue. So a neighborhood is saying that my tumor cell is next to a FOXP3 expressing cell, next to CD8 cell. We can start to look at the patterns in the composition of those tumors, and then ask the questions, how do the neighborhoods within an immune responsive patient compare to the neighborhoods within an immune sort of suppressive phenotype? And then we can sort of understand the compositions of these tumors, the different cell-cell interactions. I think it's beyond PD-1, PD-L1 at this stage. It's
right.
A lot of information we can glean from these, and potentially therapeutic as well, because if we can understand the composition of a favorable microenvironment or that of a resistant microenvironment, is there a way in which we can prevent those sort of clusterings of neighborhoods from happening, right? It's a sort of a theoretical question, but I think these are the sort of hypotheses that we can start to think about as we do a deep dive on these.
Right, fantastic. So let's talk about the technology itself. So are there any developments in these tools? Are there any limitations? So what would you say is the major limitation currently in tissue profiling that you would like to overcome, or you see us as a community overcoming in the next 5, maybe 10 years?
Yeah, so because they are new tools, the one of the challenges is the standardization, right? The data QC, the data normalization, that's all very, very new. I mean, with a bulk approach, you can normalize across the field, right? With a single cell approach, you can normalize across your different single cells. But with spatial data, the normalization of the data is a challenge. And sort of different bioinformatic teams are using sort of pseudo bulk approaches to normalize the data. But it's a challenge for the field, which is evolving at the moment. And so what I think is happening is people are generating a lot of data, but the analysis is the bottleneck at the moment, and standardizing these pipelines is one of the challenges. Another challenge is there's so many technologies on the market now. So to do spatial RNA, there's probably 5 to 10 different technologies at any one time. For spatial proteomics, it's probably the same number. So using different approaches and then pairing it with a certain analysis pipeline, that needs to be standardized across the field. So I think those are some of the biggest hurdles at the moment. And also the resolution. So the field has moved from sort of a region of interest, which is a couple cells resolution, and it's demanded single cell resolution of the whole tissue. And the challenge has been at single cell resolution, it's very noisy data. So
complexity is immense.
Exactly. And also you've got the complexity of a Z stack potentially in some of the platforms. Some of the in situ technologies, you're looking at 16 to 17 Z stacks at single cell resolution. So then you normally collapse that to analyze across it, but now you're losing data, right?
Right, because you're averaging stuff, and I mean, what happens here in the cell is not what happens here in the cell, and that's another layer of complexity.
Another layer of complexity. So these are so new tools, and the technologies are evolving so quickly that the assays aren't being standardized across the field. And what's happening is our researchers are jumping to the new shiny toy, but they're not, developing a concrete understanding of what this assay is actually, robust, reproducible, and all those sorts of things. So that's something we're working on in the background, trying to standardize assays so we know the sensitivities of them. But again, it's very new, so there's a lot ongoing, but it's exciting data. It'll probably take some time to really drill down and to be able to compare and contrast between platforms and resolutions and assays.
It kind of reminds me what's happening at the beginning with the microbiome and later in the extracellular vesicles. So everyone was jumping on the wagon and trying to do and to construct new tools and new ways to analyze, and standardization became so important. But I think in a few years it became more and more, everyone was lining up. So yeah. I think we can be optimistic about that. And speaking of extracellular vesicles, which is the main interest of the readers of this journal, and I see that you do a lot of liquid biopsies in your background. So do you see any ability in the future to put that also into tissue profiling in terms of spatial distribution? Because one of the main limitation is to see the location of which EVs are being released or being taken up on a tissue level. So I don't know where we are on that, but do you have any idea or thought?
Yeah, no, it's a great question. It's something we're thinking about in the background. Our initial approach has been characterize the tumor microenvironment, but collect the blood and bank them for EV sort of studies in parallel. We haven't gone in to do any sort of characterization on EVs in the tissue per se, but it's something we're thinking about and sort of the approaches to use there. In the blood, we can look at ctDNA and CTCs, but the EVs, they're becoming more and more important, and that's what we're realizing. We're working with a couple of colleagues at the IBN where they use some really fancy technology to capture EVs and characterize them, but it's really trying to understand the role of EVs, I think, and what that role is. It's something we're working on in the background, but which will probably take a bit longer. But we are banking samples. We are banking samples for this exact question.
Yeah, that would become really important, I mean, in the near future, but I mean, we're still not there, I guess. Let's talk about the costs for a bit. So what you do, not everyone can do, not just because of your proven skills, of course, but also because it's not really affordable, maybe. Do you see in the future how this becomes such a major and cardinal tool that it would be in every department everywhere, and people would be the main avenue?
Yeah, no, I think we was just at a meeting in The Hague last week where we were talking about exactly this. How do we democratize spatial, and how is it taken up, right? The costs are prohibitive. And even in my lab, the way we work around it is we use tissue microarrays so that we can get as many samples on a single slide, and that makes it cost effective. But the challenge is to really be able to look at whole sections, whole tissue sections. I think what will happen is as the adoption increases, the costs will need to come down. Right now, because they are sort of cutting edge, I think the companies are, it is expensive, but I think the volume of data you get back is right. It's such a, when you're looking at some of this whole transcriptome data sets that we're getting, it's over 18,000 genes per region of interest, right, across a tissue. The amounts of comparisons you can do in a well-designed study, right? You're not going to get that data set from any other approach today. So I think the costs are relative, but they are on the expensive end. But that will come down over time. But I think it's still worth the investment in that the data set you get back gives you new biology on your disease. And that's been our approach. Every study we've gone into, whether it's skin cancer, head and neck, lung, or COVID, has, it's been, when we're looking at the findings, it's always been from a sort of, what have we found here, because it's very new, right? It's a lens of discovery which we haven't had for a while. So it's fascinating.
Right, but the problem, I don't know if it's the problem, but when you have huge amount of data, it could be a blessing, but it could be a curse, because sometimes it's too much. And how do you pick, right? How do you filter what you want to present and what you want to study? How do you narrow down and put your lenses, how do you zoom in? That's
No, this is such a good question. I think the biggest thing for us is having a well-defined question up front. What is it that you're asking of the data, right? Are you looking for predictive biomarkers for chemotherapy or immunotherapy? Do your cohorts reflect that? I think that's been our approach. Very sort of basic questions. How does EGFR mutant disease compare to wild type, using a spatial lens? How does KRAS mutant disease compare to wild type? Sort of basic questions where that's what you're, and if that's your question, the comparisons you can do are defined, right? But if you go in saying, I want to do spatial and I don't know what my question is, I think you're going to get a lot of data and not a lot of answers. So I think, I think you have to be sensible in your approach. And then we've sort of gone, comparing the lungs of COVID patients to flu, comparing it to normal healthy lungs. So those sorts of questions, right, where the underlying biology will probably be dictated by the groupings that you've put together there. And then obviously you need to go validate it prospectively.
Right. So let's talk a bit about the future. Where do you see the research in your group specifically going? If we look, I don't know, 5 years ahead, where do you find yourself, like in 2028, let's say?
Yeah, my hope is that spatial scales. Right now it is very small, small groups that are doing it, or sort of R&D projects and then sort of biopharma projects. But I think what will happen is hopefully what happens is it's democratized so that it becomes cost efficient for many people to get in, generate the data set, so that, it's what single cell is today, right? It's what bulk was a couple of years ago. So everyone is using it on the disease. And I think I think that's where we'll probably get to in the next couple of years. And then really having standardized pipelines for analyzing your data sets. I think that's going to be critical. If we do that correctly, I think this has a role to potentially play in the translation, right? These are, you can discover new biomarkers associated with therapy. Obviously, you need to go validate them, but the potential to go and look at retrospective cohorts or flagship clinical trials here, where they haven't had predictive biomarkers from, PD-L1, TMB, you name it, right? We can potentially do a deep dive here on the tumor microenvironment of large cohorts when the price comes down, to really understand what's driving that resistance, what's driving that response. And I think one of the key things for us is that we can, for the first time, see resistance in situ, right? We can see the therapy resistance in situ. We can see, metabolic readout. We can see infiltration. We can see the cellular architecture. It's not just when the pathologists see these data, they get really excited because they can start to see multiple layers of data there that they couldn't see by just looking at an H&E. And I think that's going to be that flip in the next couple of years.
So also to predict, like you can see the resistance now, but could you predict it so it would become a clinical tool for an oncologist, for a physician?
That's exactly it. So if we look at, a retrospective cohort of, let's call it a couple hundred patients, we know that there was a CR and a progressive disease. We identified a spatial signature of hitting multiple things: PD-1, PD-L1, T-cell infiltration, metabolic data, multiple different pathways. And we can narrow that down into a multiplex IHC panel, which is, let's call it 20-plex panel, that a pathologist can run. Then that's useful as a companion diagnostic tool, right? So I think, at ASCO this year, the spatially linked signatures were 18-plex. So I think there is an appetite for that. And if your spatial signature is hitting multiple different pathways, then you've got something really useful there.
So how is it to convince physicians or clinicians? Do you find them they're waiting for your new solutions, or it's still hard to, because sometimes when it's too new and they would stick to what they know before they get convinced?
Great question. What we're doing at the moment, so every project, the translational clinicians are involved, the pathologists are key. So we have fortnightly meetings, are on all the calls, and they understand what we're doing, they understand the challenges, but it's also, we're digitizing pathology, right? That's that next step. So I think as long as we take them with us along this journey and don't just show an end product to them, then there will be adoption hopefully in the near future. Because you can't go to them with an assay and say, we can
this is what you have to do now.
No. Yeah, we have to take them along the journey. So that's up to us as scientists, I think, to educate the pathologists, to educate the clinicians. They can see it happening. I think we just need to make sure that they're along with us for that journey.
Right. So in the I mean, let's suppose I have no background and I'm just entering my life in research. How do I start? If I'm convinced, you convinced me that tissue profiling, spatial distribution is fantastic, and it's going to be very big. Where do I start?
It's a good question. I think so, depending on your sort of disease question, right, or your biology question, I think identifying sort of FFPE samples or some sort of sample type where you have a good comparative material there for your disease control or whatever it is, cohort study. Using a small cohort, I'd recommend running a discovery study just up front, and that's how I got into this work. I mean, we started this work with a few $1,000s. We could probably run 3 or 4 samples, but the volume of data that we got back and the information we could understand about the tumor microenvironment in cancer, in head and neck cancer at the time, was really deep. So that's what then we were able to snowball that into bigger pots of funding, to bigger grant agencies and things like that. So I think you have to start off, if your budget is $5,000, you have to start off with that. But I think the data you get back on your disease model is so comprehensive that it's hypothesis generating at the same time. I mean, you can't jump into a project and spend millions of dollars right on a sort of, not without having that initial data. So I think even with small pots of funding, you're able to ask sort of targeted questions, and that was our initial approach.
Very, very nice. So I think this was a very, very fascinating discussion about the potential of those new horizons that you are providing us, and I wish you a lot of success in your grant that you received and otherwise, of course. And I'm sure that we will be hearing more and more about this technology and about the discoveries that you can achieve with them. And I'm thanking also the journal that helped us set up this discussion. And with that, I thank you, and I hope to see you in one of those meetings soon.
Perfect. Thank you, Prof.